Thank you so much, sir, for giving this opportunity. Now I am requesting to our host, Sri Dr. Haris Gopinath Sar Garu, continue this webinar. Thank you.
Thank you, Srinath and Swati,
Thank
for
you,
a
sir.
nice introduction, and thank you all for attending this webinar. And in this, because it is very difficult to get in touch with all you people, so it is very great to meet all you people in this situation. Thank you for that also. So I'm just before going into the slide, I just want to make it as a live meeting. Like I just. I hope I'm audible to everyone.
Yes, sir.
Sorry for that. So I'm just sharing my slide. So before going to the slide, I just want to make sure that it will be like a discussion kind of thing. So it's not be one-sided that I'll be talking that you people will be listening. I don't want that. So my aim of giving the presentation is a dosage form, which is very simple to manufacture and market. That is nothing but powder for oral suspension. So PFOS, I used to call it. So we have done a lot of research work on this particular topic, PFOS, that is powder for oral suspension, or can I call it as a dry syrup. So this is very simple product, just only by taking few ingredients, by sifting and blending, we can just put it in a bottle and put it on the stability, evaluate it, and just after stability, you can evaluate the product, its performance, everything. So the product formulation part is it will be very simple, and it can be formulated within a short span of time. The problem will come when it's going for reconstitution. So powder for oral suspension is, I just want to give an introduction about this dosage form. Then I'll go in deep into the subject. So the main aim of this topic is, if I'm going to formulate a powder for oral suspension, and then I want to market in any of the regulated markets. So regulated market means it's like Europe, Canada, Australia, or USA. So what is the minimum requirement, the requirement from the agency? So that is what my topic. So you can study everywhere what is dosage form, what is the advantage, what is the disadvantage, what is the formulation aspect, what are the ingredients used in that you can see from textbook or everything. So my aim of giving this presentation is, next step, after doing a formulation, what assessor or regulator is asking or seeking in a formulation when you file into agency. So that is what my aim. So by end of this presentation, everyone should be able to understand, if I formulate a PFOS, powder for oral suspension, if I go for a regulated market, if I go for the approval to a regulated market, so what is the minimum requirement, or what is the basic thing they are expecting from us, so that we can get through the approval process? That is the thing. So here regulated, the word says that other than India, for example, like Europe, UK, Australia, anywhere if you go, we have to get permission to manufacture in India and to sell the products in their country. So in this case, we have to take a series of validation batches, like three number of batches, validation batches. So those validation batches shall be loaded into stability. And what are the documents which we did during formulation development, and then followed by doing the validation batch in a commercial scale, or a 1/10 of commercial scale, shall be evaluated, and the documents, everything will submit to agency. So agency will check, like we are submitting article to any journals, they'll check the documents, everything, and if anything doubt is there from their part, they'll ask us a query. So once the query, after they asking, we can just give our aspect of explanation for the query asked by the agency. So now coming to the point, so powder for oral suspension, when I formulate, what are the points I should consider which is critical during formulation, and as an assessor, what is their expectation on this particular formulation? So where I have to have a keen idea. So that is the basic thing, bottom line of this presentation. So I just want some people who are having doubt can raise their hands. I can have a question, or if anything during my presentation, there is no issue of that. So then and there you can ask question. And if anyone willing to answer my questions on spot, they can just also do that if anything is there from your end. So the content, yes, we'll briefly see about what is meant by PFOS, how to characterize, and what are the commonly used ingredient. Hope you will all know. As a B Pharm and M Pharm, we used to come across in our most of the pharmaceutical dosage form textbook, and preparation of how to prepare, and stability consideration, guidelines, and conclusion. So mostly I will spend most of my time in the guidelines section, where it will be helpful for you peoples. So tomorrow when you come into industry, so Sai explained, so out of 10 years of my experience, I spent 7 years in the formulation aspect. So now the day-to-day life it's easy to formulate a dosage form, but it is difficult to get agency approval. So how to get agency approval is required. So that is what I'm trying to explain over here. So there I'll spend most of my time. That is nothing but guidelines required for the regulatory agency, and what are stability testing required. Okay, so just I want to put this slide to start up. So I just want you people, if anyone can answer my question. So why to formulate drug substance into a formulation? So API is there, so I can as such give in the form of API. Why I should give in a form of formulation? Can anyone answer this question? So it will be like quite it will be interactive session, other than I can speak and you can hear. So anyone can answer why to formulate a drug substance, that is API, into a formulation? Why can't we give as such? Can anyone answer this question? So we have 72 participants. Hope everyone is listening. So if anyone can answer this question, it will be well and good.
API is not stable in our GITs.
Okay, API is not stable in GIT. Okay, maybe next.
Maybe it causes irritation.
Okay, but not most of the API causes GI irritations, or most of the are not stable in GIT. Na? So I can formulate any formula for API which is stable in GIT and not cause irritation. Can I give in directly in the form of API? It is not so, right?
Maybe release pattern also. It may be produce control sustain action.
Okay.
It may be improve the bioavailability also.
Okay. So anything else you want to guess? Bioavailability, absorption, release mechanism, anything else? Anything else you feel it is very required, sir, to formulate a drug substance into formulation? This is very, yes, for this point, I just want, yes, I want to do in the form of formulation. I cannot give in the form of API. I'm not saying no to your answers is right, but anything else, if you think it will be more apt, you can inform me. Is there anything you want to say? Anyone else? Okay. So I will just put it in this way. It's not like whatever he said is wrong. It is everything is correct. So I make formulation to make sure that patient can take, the end user is nothing but patient, can take the formulation without any issues, any adverse effect, any problem. So during taking of medication, it should be easy for me to take a medication. So that is nothing but a successful formulation. So doing a formulation successful where only patient can take, is that enough? No, I need a stable formulation. Why I need stable formulation? Yes, as per agency recommendation, we have to have ICS stability for a minimum of 36 months, 25/60, or 30/75, or 6 months, 40/75. Yes, we need stability. So is patient acceptable stability is enough? No, we also need what agency's approval for marketing any formulation. I have made a formulation, I have it is a good stable, but agency is not accepting. There is no use of the formulation. So all three things I need. First, patient acceptability. Next, it should be a stable formulation, and it should also have capability to get approval from the agency. So these three aspects, if it is there for any formulation to make into a drug substance into a formulation, that is he is a successful formulation scientist. So these three. So in this three part, everything will come in between. Whatever he said, like bio or release pattern, or this formulation will vary with respect to each and every API. So it may cause gastric irritation, whatever he said is right. So we can go into form of lymph-coated tablet. So dosage form will vary. So the first important thing is the end user should take the medication easily, or without any suffocation or any trouble. And then it should be used with it should be stable, and it should get a regulatory approval. When it will get regulatory approval? Only if it is having bioavailability, like what you said. Yes, if it is having good bioavailability only, we can get through the product. So for that, we have to formulate a formulation in such a way that it is having a good bioavailability. So these are 3 points. I just want to make sure that these 3 is very critical for any formulation scientist both before going to the thing. So as a general introduction, I just want to give dry powders for oral suspensions or powder mixture. The required addition of water, reconstitution at the time of dispensing, and are mostly for pediatric use. These are called dry syrup or reconstitution oral suspension. So this is a textbook definition. I will just put it in another way. Whenever the API is not stable in the form of aqueous solution or suspension, so I cannot make a formulation into a liquid dosage form, but to give into a pediatric purpose, especially pediatric purpose. Why? Yes, children cannot take these medications. It will be very difficult to take a tablet or capsule. It may cause choking, means it will cause choking to the throat. So in that case, what I can do? Yes, liquid dosage form is always a friend or a best dosage form for a pediatric, for a children's. So for those children's, I want to give a medication, but whereas my API is not stable in the form of aqueous solution. So what I'm going to do right now, I will take the API, I'll mix with some excipients, and I'll make into a formulation that is nothing but powder for oral suspension. Means what? This dosage form will remain as a dry powder till usage. So once it starts, the day I want to use, I'll reconstitute with water, and then I will start taking the medication. So this is what PFOS means. So throughout the lifespan of the dosage form, it will be in a powder form, which will be filled in a bottle. And then when I want to use it, the medication, I will go for reconstituting with water, and then to the required amount of water, which is given in the bottle. And then I'm going to take the medication. This is nothing but dry powder oral suspension. By this way, what I can get, so first thing is the stability of the formulation can be improved. I can give more shelf life. Means what? If a formulation is not stable, API is not stable in aqueous, I'm giving it 6-month validity, shelf life. But rather than I'm giving in the form of powder for oral suspension, where I'm giving here 2 to 3 years. So I can improve my commercial and also my market feasibility. So sales will be more. So that is also should be a consideration during a formulation. So by this way, I can improve the shelf life of the formulation by giving the form of PFOS. So 2 PFOS are there. One is single dose. Just you reconstitute it, take it 1 dose over, we can throw the medication, or we reconstitute, we take for 7 days or 10 days or 14 days based upon the pill which is mentioned in the bottle. So we used to take, right? So from child, we used to get some medication. So once you reconstitute it, doctor will say you keep it only for 1 week or 10 days in a fridge or in room temperature. After 1 week or 10 days, you should be thrown. But if it is not reconstituted, yes, we can keep it 2 to 3 years also. So this is the basic use of PFOS, where most of countries like Canada, Europe, they are targeting this dosage form because yes, pediatric is one of the critical formulation where we have to take each and every step very cautiously, because we are going to dose for a patients which is age of less than 12 to 15 years. So rational, inadequate chemical stability, as I explained. If API is not stable, yes, I have to go in the form of PFOS. Avoid physical stability problem like viscosity changes. If you see we are using xanthan gum or sodium CMC, these are nothing but gums which has tendency to swell or increase the viscosity during storage. During storage of increasing viscosity, what will happen? Sometimes the product will not come from the bottle, so it will have a flowability issue, and then the patient cannot be able to take the medication. So these problems can be overcome by giving the form of PFOS. And then polymorphic form conversion. Yes, any API, if it is there in the form of solubilized form, in the form of solution or something like that, chance of conversion of polymorphic form is there. Means change of one form to other form, crystalline form of one form to other form. An incompatibility issue and crystal growth, caking is also other problems of giving of PFOS in the in the form of solution. Reduce the weight of final product, means instead of giving in the form of syrup, which is always heavy. So PFOS is nothing but dry powder. So it will not have any water. So carrying is also easy. Whenever I want, I'm going to reconstitute, I'm going to take. shipped without regards to seasonal temperature. So I will not worry about condition during shipment. So if it is there somewhere around 25 degree, I'm not going to bother about the condition during my shipment. If I want to manufacture in India, I want to send it to Europe. So I'm not going to bother more about the shipping condition because it is in the form of dry powder and it is highly stable. So generally, I just want to classify PFOS into two categories only. One is sugar-based, which is having a sugar as a diluent, and second one is sugar-free. It is nothing but using a mannitol or sorbitol as a diluent. So two basic type of PFOS. So one is sugar-based. So sugar will be the major constituent which you use in our daily life, and then sugar-free is nothing but other than sugar, which is not used here. It will be mannitol or sorbitol. So I just given an example of some formulation is just for your understanding. So I just differentiated what are the excipients will be used in a PFOS, powder for oral suspension. So here, what are the excipients I'm using? API, that is nothing but amoxicillin trihydride. It is a beta-lactam antibiotic. I am using a sweetener. I'm using a suspending agent. I'm using a desiccant. I'm using some buffering agent. I'm using some preservative. I'm using some colorant and flavors. So these are the basic or minimum requirement of ingredients which are used for a PFOS formulation. So I just given some example. If it is sugar based, sucrose I'm using. If it is sugar free based, I'm using mannitol. If it is a suspending agent, yes, for suspension I need suspending agent to make sure that it is not forming a cake or it is not going sedimentation. So that I need this suspending agent, xanthan gum or sodium CMC. Desiccant is nothing but silica gel, is nothing but xyloid derivatives which used to observe the moisture during the storage of the API, and it will not allow the API to expose to the moisture environment. By that way, it will improve the stability of the drug substances. Preservative, obviously, sodium benzoate or other preservatives is required because during storage or after reconstitution, a period of 10 days or 14 days, if any chance of contamination with microorganism is there, so we have to go with sodium and benzoate. And it is not only act as preservative, it also have a tendency to improve the stability of the API. For example, amoxicillin and the clavulant potassium, most of the dosage form will have sodium benzoate as preservative because this preservative have tendency to stabilize the API. By that way, decrease the rate of degradation of that is our drug substances, and then increases. It is make sure that it is stable and it is not increasing the RS residual. Radius substances will not be more if you use sodium benzoate as a preservative as well as stabilizer in a PFOS formulation. And as to make it attractive, we are using colors, and to make it more pleasant or palatable, we are using flavors. So these are the general ingredients which will be used during a PFOS formulation. So this may vary with respect to one formulation to other formulation, but most of the things will be there available in any pediatric to make sure that it is attracting the child and it is easy to take. It is not having too bitterness, so we are using a sweetener, artificial or whatever it may be, and we are adding a color to make it more attractive. We're adding flavor to make sure that whenever we take the medication, we feel pleasant. So this is the basic PFOS formulation ingredient available. So what are the characteristics required for suspension for reconstitution? Okay, so first, powder blend must be uniform mixture of the appropriate concentration of each ingredient. So what I want to say here, whenever I make the formulation, so it is very simple step. First step is I will take all the ingredient, I will pass it through suitable mesh. I will then that's nothing but sifting. Next step is nothing but after sifting all the ingredient, I will put it to a blender and will blend it for X or Y minutes, and then I will take out the blend. That is the final blend, and then I can manually fill, or by using a machine I can fill into a bottle. So this is the step. So for sifting, next blending, then bottle filling. This is the basic 3 steps which is involved during manufacturing. Now in any formulation, yes, we have to ensure the drug substance uniform distribution. That is nothing but API. I will take example of amoxicillin suspension. So as we given the example of amoxicillin, I will take same amoxicillin suspension as an example. So I have a bottle. So I'm using a multi-dose container. So can anyone briefly differentiate between multi-dose container and what is a single dose unit dose container? Any difference? What is the major difference in that? Single dose and multi-dose container? Is there any difference between the single dose and multi-dose container?
Yes, sir. There is a difference, sir.
Yeah, what is the difference?
Sir, it can accommodate only
Yes, correct. You're right.
one dose. That is called
Mhm.
single dose. If many doses can accommodate, that is called multiple doses.
Okay, so answer is there in the question only you are saying. So a single dose means it is only one dose, and multi-dose means. So what is during manufacturing? So we will ensure that it is a single dose or multi-dose based upon the weight. And if it is multi-dose, for example, 20 doses are available. 20 dose means what? 20 times I can take the medication from the bottle. Is that right? So 20 dose means 20 times I can take the medication. So what is the per dose? So every PFOS will have the label. Each 5 ml contains, each 1 ml contains, means what? So one dose is nothing but 5 ml or 1 ml. That will be written clearly in the medication. So like that, if each 5 ml contains 125 mg of amoxicillin means, then 20 dose of amoxicillin if available means, then 20 into 5 is nothing but 100 ml deliverable volume will be there in each bottle. Means what? 100 ml is total volume. Each 5 ml contains 125 mg. So like that, 20 doses are available. That is nothing but multi-dose container. So this is what difference between single dose and multi-dose. If it is single dose, yes, for the parenterals we used to use, only one dose will be there. Just break the container, add some parental saline, and then shake it and directly inject it. After that, there is no use. So it will be directly discarded. But for oral purpose, we are using is a multi-dose container where it can be used for 20 dose or number of doses for so many days, X or Y days. So they will be given particular temperature and days, how we can store, where you can store, how many days you can use. For example, there are some formulation which can be stored at room temperature also for 7 days. So that can be stored. But there are some formulations which cannot be stored in room temperature, like potassium clavulate. It is having chance of high degradation if it comes in contact with heat. So room temperature is not sufficient. So we have to go with refrigeration, means keeping in a fridge. So if keeping in a fridge, what is happening? So I can extend the shelf life of reconstituted suspension. So I am giving dry powder 3 years as shelf life for dry powder. But once you reconstitute, it is only 10 days. So once I reconstitute, it is nothing but 10 days. After that, even if there is some amount of dosage form available after reconstitution, you cannot use, because the assay of the formulation will be coming down and there won't be any potency in the formulation. That is why, so we are giving 3 years for a dry powder and only maybe a week or 10 days for reconstituted suspension. Okay. So here, what is the concern? So sifting, blending, and mixing is nothing but the 3 steps involved in the manufacturing of PFOS. So first, during reconstitution, the powder blend must disperse quickly and completely in the aqueous vehicle. This is very, very important, because we have a powder, we are adding some water, and I'm shaking it for half an hour. Is it a good formulation? I'm damn sure that it's not a good formulation. Why? A patient should take a medication who is suffering from some illness. So your formulation should assist them, not to make them worry. So here, reconstitution time is very, very critical. So we thinking, so here in India, we used to get the medication from pharmacy and we used to reconstitute. But for example, if I'm going to Canada or Europe or USA, the pharmacist will reconstitute the suspension and they will instruct the patient how to take the medication, how many number of days to have take, and everything the pharmacist will insist to the patient. So in that case, if a pharmacist is reconstituting your suspension, which you have formulated here, and you are from India, he is from Canada, he is trying to add a water to a bottle, and then he is trying to reconstitute. It should be within a span of 1 or 2 minutes, but it should not be more time. If the more the time, and he will think the formulation have some issue with the stability, and he will say this formulation is not good, you can take back to your India. So that is, reconstitution time is very, very critical. So that is what I'm saying. During reconstitution, the powder blend must disperse quickly and completely in the aqueous vehicle. So this is one of the very, very important regulatory aspect from any agency, reconstitution time evaluation, how to do. So that I'll explain in next coming slides. Next, reconstitution suspension must be easily disposable and poured by the patient to provide accurate and uniform dose. Means what here I'm trying to say. So if, for example, I said a bottle contains 20 doses, means each dose is 5 ml. So if I take today two doses, so I have to take the dose for 10 days. So I have taken 9 days, 2 doses I have completed. So as I calculation, 18 doses completed. Then I'm coming on the 10th day. Morning I have taken 5 ml. Yes, completed. So one more dose. On 10th day, when I want to take the second dose, I'm seeing the container is empty. So as per label claim, we have to have 20 dose. That is nothing but 100 ml. But when the patient takes, he is taking every day 5 ml by seeing the cap. There will be marking on the cap. So yes, day 1, day 2, day 3, every day he have taken properly. And day 9, he or she taken properly. And day 10 came. And day 10, after taking the 19th dose, 20th dose is not there. So it is went, it is just not went like that. So it is problem with deliverable volume. So when a formulation you're doing, how much amount of reconstitution water? So if I take a bottle, if there is a powder to a certain extent, how much ml of water I have to add into the powder bottle so that the final volume will come 100 ml. This should be properly evaluated during our development studies, or otherwise there won't be sufficient dose to deliver the actual number of dose. For example, if 20 dose should be there, now the patient is having only 90 dose. It is because improper evaluation of reconstitution volume. So the container, how much amount of water I am adding is nothing but reconstitution volume. I'm shaking for a certain amount of time, that is nothing but reconstitution time. And after reconstitution, how much ml came, I'm just transferring into a measuring cylinder, that is nothing but reconstitution, deliverable volume, final volume, that should be minimum of dose what is available in per bottle. For example, if 20 dose are available, the minimum volume should be 100 ml. So that should be properly evaluated during our development studies. Final product must have an acceptable appearance, odor, and taste. Very, very important, because if any of this particular appearance, odor, or taste is not palatable, appearance is not good, if odor is not acceptable, and taste critical for any beta lactam, because amoxicillin and most of the formulations are, it is highly bitter in nature. So if you are not able to mask the taste of this bitter API, your formulation is not going to succeed in the market and it will come back to you. So these are the 3 very, very critical parameters: appearance, odor, for that we are adding flavor, flavoring agent, and taste, by we are adding a artificial sweetener or natural sweeteners available in the market. So commonly used ingredient, so you would have come across these ingredients: suspending agent, wetting agent, sweetness, preservative, flavoring agent, buffering agent, color. And infrequently we are adding anti-caking, flocculating, solid diluent, anti-foaming agent, granular binder, granulate disintegrant, antioxidant, and lubricant. So this is frequent means, yes, 90% or 99% it will be there in the formulation. And infrequent means 1% chance is there. If there is any problem with a particular API, yes, this 1% chance of adding infrequent ingredient possibility is there. Okay, so difference is there: frequent, infrequent. Frequent means all the PFOS formulation 99% will have these ingredients. What are that? Suspending agent,
suspending agent, flocculating agent.
Fine. So this is mandatory. So most of the 99% of formula, without this I cannot make a PFOS or a suspension. So I can, but infrequent is, if there is any problem with the API or formulation, to overcome that, I'm going to add infrequent ingredient like anti-caking, flocculating, solid diluent or antioxidant, whatever it may. Okay. So number of ingredients should be kept minimum. So why it should be kept minimum? Always the formulation should be very simple. Simple in the sense, what is required for the formulation? It should be stable. It should not undergo sedimentation. Even if it undergoes sedimentation, it should be easily redispersible. It should be pleasant. Color should be, appearance should be good. Taste should not have bitterness. So you have to have some sweetener, and then flavoring should be there. Means this is basic, right? So what is the basic? That only you have to give. I'm trying to do more. If you try to add other extras extra in the formulation, that will try to give you a product which is not having good quality. So that is what I'm saying. Number of ingredients should be kept minimum. All ingredients that perform more than one function, sucrose. So sucrose here, what is the use of sucrose in a PFOS? Can anyone say?
Sweetening agent, sir.
Okay, only sweetening agent. Is there anything else? So I'm saying like more than one function means water. So here there are some ingredients which I can use for more than one function. Sucrose, as you said, it is sweetening agent. If it is sugar based, I'm using it as diluent. Diluent means water. I'm adding all the ingredient. After adding all the ingredient,
to get a large amount, we use diluent, sir.
Yes. So here what I'm trying to say is, If I have a formulation with sucrose based, okay, here for my bottle weight is 30 gram. So after adding my API, after adding my wetting agent, after adding preservative, after adding flavor, buffering agent, coloring agent, my total weight of the bottle is 10 grams, it's coming. So what I'll do, the 20 gram remaining, I will add sucrose. So here I'm using sucrose for two reasons. One as diluent and one more is sweetening agent. So two function, one ingredient will always have a good impact rather than adding more number of ingredients. So it is nothing but making your formulation effective and simple. Understood? So all ingredients should be dispersed rapidly on reconstitution. Very critical point. If you're adding any ingredient which is not dispersing quickly in a suspension formulation, you should not consider that for a PFOS formulation, because this is very critical step. And you cannot say, no, sir, I'm adding this because it will be very stable. Formulation will be stable, but your formulation is a powder for oral suspension, which should undergo rapid dissolve when it is coming in contact with water. So if you are not adding a proper or correct ingredient, it will cause a problem to the formulation. So these are the typical reconstituted oral suspension. So having amoxicillin trihydrate, so ampicillin, so all beta-lactam antibiotics. If you see, kids are prone for fever, all those things. So we used to give all the antibiotics beta-lactam and dicloxacillin, cloxacillin derivative, penicillin derivative, ampicillin, everything is nothing but antibiotics. So which is used in the form of PFOS. Why? These antibiotics are not stable in the solution form. So to improve the stability, I have to formulate in the form of PFOS. And to make it easy for administration to kids, I have to formulate in the form of PFOS. And to increase the marketing availability. Marketing means what? Instead of going for 1 year shelf life, I'm going for 3 year shelf life. My market ability is more. I can make it more income from that particular product. So even now also PFOS is having a large volume, large income in today's sectors also. So nearly all drugs formulated as reconstitutable oral suspension or antibiotics. Beta-lactam antibiotics is all PFOS in the form of PFOS. Sodium dicloxacillin is water soluble. It is formulated as an insoluble form in suspension to help mask the odor and taste. Critical, very, very critical is taste. Based upon the bitterness of your API, your formulation should be more specific to target the problem. So if it is amoxicillin, I don't have problem with the molecule for stability, but I have problem with the molecule for taste. So taste masking is important. If it is amoxicillin with potassium carbonate, I have more problem with stability of the formulation, because potassium carbonate is not stable at heat. If it is coming in contact with moisture, it undergoes degradation. So always I have to consider based upon the API I'm using. For that we have to give a proper solution. So suspending agent. So all these things, I hope you know, acacia, CMEC. So these are suspending agents used, xanthan gum, tragacan, silicon dioxide. Silicon dioxide colloid, we have different grades. We have fluffy grade. So many grades are available for PFOS to use to absorb the moisture. So those things are there. Suspending agent, yes, easily dispersed by vigorous shaking, by shaking during reconstitution. Combination of MCC and sodium CMC is a common suspending agent used with the reaction. So I just want to go to my main topic. So this is just a general way of preparation. Powder blend, granulated product, combination product. So it's nothing but powder blend means, as I said, simple process: sifting, blending, and bottle filling. Granulated product, I will go for RMG. I will take granules, and then I'll go filling. So ingredient of the dry mix powder form. Ingredient present.
అదే రెండు ఉన్నాయి కదా అది వెళ్ళిపోయిందమ్మా ఇప్పుడుగా వెళ్ళిపోతాయేమో అవి.
So mixing ingredient of dry mix ingredient present in small quantity may require a two-stage operation. So the powder blend, sifting and mixing. So it is least capital, means equipments, very simple equipments are required. So chemical stability is more. So exposure also will be less. So it easily can be formulated. Prone to homogeneity problem, means uniformity is very, very critical for sifting blending process. That is nothing but homogeneity issue. That is, uniformity of blend will not be available. And particle size and powder flow, loss of active ingredient during mixing, potent drug used in very low concentration is critical. So sifting and blending, segregation is only one problem which you have to tackle. So during blending or sifting process, chance of segregation is there. more variation in the assay is there during blend stage or reconstitution stage. So that is the one critical we have to evaluate during a process by using powder blends. So granulated product, very simple wet granulation process. So we'll take up API and other excipients in the wet granulation RMG, and then we'll make into a mass by aqueous or non-aqueous binding solution. Drugs can be dried, dry blended with other ingredient. It can be dissolved or suspended in the granulating fluid. So solid layering or blending and masked with granulating fluid is a planetary mixture. So here nothing but, instead of, if in case flow is not good for the powder blend, so I want to improve the flow. If my API is bulk, so bulk density of the API is very less. I want to make it more flowable so that my bottle filling will be very useful, or it will be very easy. In that case, yes, I have to go in the form of granulated product. It's nothing but making the form of granules, converting the API into granules by adding some binder, and then add the other ingredient like flavoring agent, coloring agent in the extra granular part. Before blending or during blending process, I can go for bottle filling activity. I hope I'm audible and my video is clear.
Yes, sir.
Yes, okay, sir. So this is nothing but granulated product. So combined product is nothing but it is having one or two ingredients. So I'll just go through the advantage of granulated product. So improved appearance, improved flow characteristics, very, very critical. Flow characteristics, if flow is not there, uniformity also will not be there. So in the case of low bulk density products, we have to go with granulated product, which can improve the flow property. Less segregation problem. So less segregation problem, less generation of dust during filling operation. So during filling operation, dusting is very, very, very critical. Why I'm saying, if it is in the case of powder blend, only sifting and blending process we are going, we cannot control the percentage of fines which is there in that process. But in the case of granulated product, we can control as we are going for granulation approach, where the percentage of fines can be reduced to a level based upon the amount of binder we are adding in the granulation process. And by choosing the excipients particle size, the dusting can be reduced, because dusting is always a major concern during a bottle filling approach. So here more capital required and energy is required, as you know that we need a RMG everything, and it is more energy and more time required. Difficult to remove the last traces of granulated fluid, reduce the stability. Uniform granulation is necessary. Excess of very small particles or fines will result in a rapid segregation. So particle size ratio is very, very critical for granulated product. So there in the powder blend, we are not doing any process, just only sifting, blending. So blending time optimization we have to do to make sure the uniformity. As we are here, we are doing in the granulation, we are adding the binder into RMG. We have to make sure what is the target PSD we want. What is the 60 mesh? What is the percentage above the 60 mesh retain? What is the percentage below 60 mesh retain we want? By evaluating this only, we'll come to know, is it going to affect the flow property of the final blend? So that is very, very critical, as it is involved some change in the process. So granulated product PSD is very, very critical during formulation. So here it is combined product. So less energy and equipment for granulation may be required if majority of the diluent can be added after granulation. Heat sensitive ingredient. Yes, clavulant potassium is highly heat sensitive. So we cannot go formulation by normal way. Such as flavors can also be added after drying of granules. So first to granulate some of the ingredient and then remaining ingredient. So first one part we are made into a form of granulation. We increase the flow property of the blend. Second step, whenever API is available, which is having a heat sensitive, I cannot go for granulation. Then that ingredient I will add after granulation of some part of the diluent or some of part of the excipient. Then I will add it in the extra granular part. The heat sensitive material like flavor or API or whichever is required can be added in the later part of the formulation. So risk involved is nothing but the risk of ununiformity. There is nothing but distribution issue. Particle size of various fractions should be carefully controlled. Here controlling of particle size is always critical, because initially you are going for one process and then you are later a second part you are going for other process. So chance of variation in the PSD is always there. So which should be controlled. So advantages and of different types of mixtures. So powder blend, economic, low incident of instability. So because there is no contact of any other material other than what you intended to use. If I have 5 ingredients, only 5 ingredients are there in the process. Only sifting, blending and bottle filling. So that is the economic process, and it is having no moisture or no chance of exposure to other ingredient or material outside this. Mixing segregation problem is there. Yes, we have to optimize the blending time, and loss of drug is always there because we are adding the form of dry form. So more fines generated, chance of loss also more. So careful observation required during manufacturing. Granulated product, appearance, good flow property, less segregation, less dusting. Chol is a characteristic improvement of granulated product. Cost, yes, we have included cost is more because time of manufacturing will also be more. Effect of heat and granulated fluids on drug substance and excipient, chances there. So we have to control that. Whichever excipients is undergoing degradation in that heat condition, we have to omit it during granulation, and that material can be added in the form of combination products. So reduced cost, use of heat sensitive ingredient can be used by combination product. So here ensuring non-segregation mixture of granular non-granular ingredient can be maintained by PSD evaluation. So recommendation. So what is the basic recommendation for dry mixing? Use effective mixing. Means what? Blending time optimization. So in blending time, there are two parameters. One is blender RPM and time. So whatever the RPM and time we are keeping here, we have to optimize it to give uniformity of dosage material. Determine adequate duration of time, that is mixing time is nothing but blending time. Avoid accumulation of heat and moisture during mixing. So the environment and the internal environment during the manufacturing process should be controlled to avoid any absorption of moisture to the blend. Limited temperature humidity. So here I given temperature 70 at 40 degrees Celsius. So it is not 70, sorry. It is temperature 25 plus and 40% RH. Sorry for that. 20 degrees Celsius, so 25 degrees Celsius and 40% RH. Finished batch should be protected from moisture, as most of the beta-lactam are sensitive to moisture. We have to always cover the material. So the area should be, relative humidity of the area should be less than somewhere 30 or 20. Sample for batch uniformity. So the batch uniformity sample should be taken from different locations. By that way, uniformity can be evaluated. Stability. Physical stability should evaluate both dry mix and recon suspension. Common evaluation of recon suspension includes sedimentation volume and easy of redispersion. So easy of redispersion, nothing but redispersibility of any particular suspension. Exposed to a cycle of temperature change, freeze-thaw method. So if you request a patient to keep in fridge for 10 days, so your formulation should be stable if I keep in a 2 to 8 degree also. So if I keep in 2 to 8 degree, your formulation should remain stable and should not be converted into a solid mass. For example, we have seen some of the things if we keep in the fridge at 2 to 8 degree, it will convert it from liquid to solid mass. That should not happen if you insist a patient to store it 2 to 8 degree. So chemical stability is always critical, should be determined in both the dry mix and recon suspension. So agency recommendation to nowadays, we have to prove the dosage form is stable not only in dry mix. During stability, it is in dry mix form, but when the patient takes for 7 days or 10 days, it is in the reconstitution form. So we have to provide justification and data stating that both dry powder and recon suspension is also stable throughout the period of manufacturing or shelf life of the product. So both should be explained not only at controlled temperature, but also potential exposure such as during shipment or storage of the product. So we also should evaluate the extreme condition. So what is the maximum condition it can withstand the formulation? Maybe 40-75 or 30-75. What is the condition where it can withstand? So that how many months we can withstand, we can evaluate, so that the pack, with respect to what pack we can use, it can be HDP bottle or glass bottle, can be evaluated for PFOS for a particular market. Stability evaluation of reconstitution oral suspension should be conducted in a container of the same material and the size in which the product is marketed. So what is I'm trying to say is, if you are evaluating in a glass bottle throughout your formulation, but when you go for a marketing, you want to reduce the cost. So glass bottle compared to glass bottle, if you feel HDP is very cheaper, and during marketing, if you change the marketing pack from glass bottle to HDP, if you're not having sufficient stability data, not possible. So whichever the data you're generating in your development study, so the same pack should be manufactured in the validation batches also, and for that only approval you will be getting, and for that pack only you can go for commercial. It's not that today I can manufacture with glass bottle, tomorrow I'll go with HDP bottle, sir, because cost is different is there. So it will be very cheaper. This is not possible. So if you feel like HDP is cheaper, glass bottle is costlier, then you have to load both the condition and you have to show, yes, in glass bottle also it is stable, HDP also it is stable, yes. So that what happened? I can go for marketing in any pack. So the agency also will accept either in glass bottle, HDP bottle. As you, so your given data is good, I am giving grant for both the pack. It is not like doing here one pack and marketing another pack. That is not possible. So effectiveness of the preservative is determined by challenge test. During products are often exposed to elevated temperature for the determination of shelf life. So that is 4075 condition, accelerated stability study for 6 months. If you evaluate, if a product is stable for 6 months, it states that it will be stable at 2560 degree for 36 months or at least for 24 months. So you can get minimum shelf life of 2 years or maximum shelf life of 3 years. So here where I just want to spend some time and ask you people more questions. So what is a critical test when it comes to PFOS? So as an agency, what is the requirement from agency? What they are trying to ask? And here, what are the tests we have to critically evaluate in a PFOS formulation? So as we know, we have to do assay. Suspension, as the API is not in soluble state, we have to do dissolution. We have to do RS related substances. Other than these three things, other things what is critical is pH. So why pH is very critical? pH is one of the parameter which is evaluated after reconstituting. What is the pH during the stability? I'm going to fix. So monograph, most of the antibiotics having monograph. If monograph is giving 3.8 to 6.6, I'm going to keep the same limit. What will be the problem of keeping a pH range? Here, why it is critical means pH range of monograph is a broader limit, 3.8 to 6.6. But agency will ask us what is the data generated by you stating that throughout the pH range your product is stable, because most of the condition we are using sodium benzoate. Sodium benzoate is not stable above pH 5. So if it is not stable, there won't be preservative effect. So if it is not having preservative effect and your formulation is having a pH of around 5.5 during 10th ray or 14th ray, then if you don't have sufficient stability data in that condition, agency will not accept your formulation. So what I'm trying to say is monograph pH range is for our information, but we have to generate data for pH data for whatever the range we are studying. If you are supposed to keep like 4 to 6 is the pH range I'm proposed to keep in my product, in the pH of 4 to 6, your product should be stable and should be all the parameter, assay, RS, dissolution, all the parameters should be consistently reproducible. If it is that case, only the agency will give permission. So pH is very critical. Next, chemical analysis of drug and preservative. So we are adding preservative. So as we are adding preservative, we have to do evaluation. So normally we used to do assay. So only assay is enough, or any other test is required for evaluating preservative? Is there any test available for evaluating preservative? So it is question to the audience. Is there any test available for evaluation of preservative? Like sodium benzoate or whatever you're using in the formulation, is there any test available in the market?
We don't know, sir.
Sorry, I'm not able to get you. Sorry.
We don't know, sir. Exactly, we don't know, sir.
Okay, exactly, you don't know. Okay, is it you are thinking, is there any test will be available? Any chance of guessing? So you may not know, but you can just guess, right? Because if we are doing assay is nothing but water. How much amount
Analysis.
of yeah, sorry.
Analysis the quantity.
Yeah, analysis in the quantity. So now I analyze the quantity. For example, I'm adding 5 mg per 5 ml. So I'm doing the analysis and I'm saying that yes, 99% assay I'm getting. So it says that sodium benzoate preservative is available. Will that be sufficient, or will something else also required? I'm showing 99% assay coming during stability or during initial analysis, everywhere 99 coming. It is uniform. So what else required for a preservative to be evaluated? It's very, very critical. So there is a test called preservative efficacy test. Okay, PET, preservative efficacy test. So without doing this test, you cannot market your formulation stating that, example I'm telling, now I am using 5 mg per 5 ml. So you are using 5 mg for, how you are saying that it is effective in your formulation. So for that I will do PET study, preservative efficacy study. So not only in 5 mg, I will first give placebo. I'll give 1 mg, 2 mg, 3 mg, 4 mg, 5 mg, 6, 7, 8, 9, 10. So different concentration of sodium benzoate, I'll give it to the PET study evaluation. So during PET study, placebo will have growth of microorganism. Study will be done for 28 days. So nothing but you will take a broth, you will inoculate some amount of microorganism. So then I will put my product. So log reduction it will give. Log reduction nothing but example I'll tell in a raw way. 1000 organism I'm inoculating in one media. So 1000 organism is there initially. Now I'm giving my placebo formulation. So the 1000 organism will be there throughout my study. So there won't be any reduction because it is placebo. Second, so which is having API but not having sodium benzoate, because API is already is a antibacterial. Antibiotic is nothing but antibacterial. So it will have tendency to impact the any microorganism. So I will give only formulation with API without sodium benzoate as preservative. Then third, I will give with API with different concentration of sodium benzoate. Second thing, without API with this, sodium benzoate at different concentration. By this way, I will say that yes, I've evaluated all this concentration. This particular minimum amount of preservative required for my formulation to make sure that no growth of microorganism. For saying that for this preservative we need PET, preservative efficacy test. So this test will be always will be done in the industry before going for a validation batches. So we'll say how to optimize the formulation. Now if I say 5 mg is required, tomorrow the other person will come, no, 10 mg only required. No, it is 2 mg also enough, 1 mg also enough. So it can be case to case varied. So for having a conclusion for this, we have to do a preservative efficacy test, and based upon the preservative efficacy test outcome, I will finalize my quantity of sodium benzoate required every 5 ml. Either it is 5, 2 or 1 based upon the study result. So it's very, very critical. Other than assay, we have to do PET study. So that is preservative challenge test at the initiation and conclusion of the study. So I have given two initiation and conclusion of the study. What is the difference, ma'am? Initiation and conclusion.
Initiation means starting procedure, sir. Conclusion means from all that what we get.
So I don't think so, because I'm saying in the other way, initiation and conclusion is nothing but initiation may be initial period of study. So my product is fresh, today I made. So initially I'm doing it. Now I'm giving you the sample, you're evaluating it. That is initial period of study. After that, this product is loaded in stability, 40-75, 25-60. So conclusion of the study is nothing but during end of my shelf life. So 36 months, if I say also, at the 36th month, for example, 35th month sample, I have to remove just one month before. I will take the sample, I'll go for again PET study. Why? Initial time, your preservative is effective. But what about the end of shelf life? So that also should be evaluated for a PET, for a sodium benzoate efficacy. So that is nothing but initial is nothing but a new batch. When you take, that is initial. And conclusion is nothing but end of shelf life. So during the end of shelf life also, the preservative efficacy test to be conducted. Next, appearance compared to that of sample stored at 2 to 5 degree. This is freeze thaw. So if you say that 2 to 5 degree or 2 to 8 degree or formulation should be stored, appearance should be, if you have it is a milky viscous suspension, there also in fridge it should be milky viscous only. It should not be formulation of some lumps or viscous, or it is not possible to pourable. It should not be there. In that case, you have to reformulate your formulation. So next, coming to viscosity. Okay. So viscosity, my product is only dry powder. And whenever I want to take the medication, I'm going to reconstitute it. I'm using only for 7 days or 10 days. Is there any criticality of this viscosity in my formulation? Because throughout my lifespan of my product will be powder. It is not going to have any viscosity because there is no viscosity for powder. So during administration, because I say today I have disease, today I'm reconstituting it. I said I am going to take for 7 days or 10 days. Is there any critical or viscosity is very, very critical?
I think not that much, sir.
Okay.
Viscosity that much not affects.
Okay. So I am saying it is very critical. Why? Because I'm using suspending agent. Suspending agents are what? Can you give some examples?
Methyl cellulose.
Okay. But in this case, I'm using xanthan gum, sodium CMC. Okay. So these are suspending agent which I'm using formulation. So these are natural gums, right? Natural gums, what is the tendency? On storage, after reconstituting, slowly it will get swelled. After swelling, what will happen? It will try to increase the viscosity of the formulation. So what happened? The exact quantity of suspending agent added into a formulation or not, how to evaluate? Now viscosity will give the answer for that. Why? If I am, for example, I'll tell, I have added one formulation 500 mg, one formulation I had 250 mg, one formulation I had 100 mg. So 3 formulations is there, 500, 250, 100. So I'm after reconstitution, I kept on my table RT, room temperature, I'm kept. Initial, I'm checking viscosity for all 3. So 500 should be around 1000 viscosity, 1000 CPS. 250 is around 500 CPS. 100 is around 200 CPS. So exactly double. So okay, fine, no issue. Again, I'm checking on 7th day. So the 1000, whichever is there on initial day for 500 mg. Now on 7th day, what happened? It has become 5000. Okay. And next for 250 mg, what we kept the sodium CMC, that initially it was somewhere around 500 CMC, it was around 250. Now it became into 500, 600. It is nearby. Not drastic increase, slight increase, but it is nearby. And next third, whichever added 100, the 200 CPS of viscosity. Now again I am checking, it is also again 200. No change in the viscosity. What it says? So the optimization of suspending agent is based upon viscosity. So the viscosity, if it is not increasing for 7 days or 10 days or 14 days at RT condition or 2 to 8 degree condition, it states that the solution is not getting viscous, so that it can be easily poured out of the container. So which is very critical. Removal from the container, content of the drug should be easily removed from the container. For that, what required? Pourability. If viscosity is slightly increasing, sorry. So viscosity goes on increasing. So we don't know at 36 month, now it will be dry powder, no issue. Then after that 36 month end of shelf life, I'm taking one bottle reconstituting, it will form a gel-like mass. And will that be acceptable? No, right? So viscosity can be evaluated during the formulation development. So compare, is there any increase in the viscosity during my period of after reconstitution? So if it is increasing, is there, how much increase is there? Is it acceptable or not? So I have considered myself, if 80 to 120% change is there. If you consider average 80 to 120% change here or there, no issue. If there is drastic increase more than that, it is critical. So viscosity is always plays a crucial role for a flowability of suspension. So it is also should be evaluated during a formulation. So homogeneity is nothing but content uniformity. So I have made a formulation, filled in a bottle, reconstituted it. So suspension is nothing but sedimentation chances there. So we have to always shake well before use, we used to give. So after shaking, we have to ensure the content is uniform. How will you ensure? Top of the bottle, middle of the bottle, and bottom of the bottle, you will take 5 ml of the sample. And then you will give for evaluation of assay. After reconstitution, you used to do assay, right? So that we have to do same for here also. Why this is critical? Nowadays, agency is asking for more uniformity related questions. Means what? You did a formulation. We have shown in dry powder, blend uniformity evaluation, blend RPM evaluation we used to do, and we used to give the data. So every 10 location is very good. Composite assay is very good. Everything is good. But what about reconstitution suspension? That is the end product. How he is going to use. A patient is going to use not dry powder. He is going to use a reconstituted suspension. So we have to evaluate from top, middle, bottom. So we have to evaluate and say that yes, my top, middle, bottom is also having same amount of API as given in the finished product or as given in the shelf life specification. So we have to give that consistency batch to batch. So if this consistency is not there, your process, your manufacturing process or flow is not correct or not consistent enough to give a robust formulation. So that it can be evaluated by content uniformity. Content uniformity is nothing but assay. So we used to do bottle to bottle. So not here bottle to bottle, we are doing different location of the bottle. So I used to do 10 bottles, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 bottles we used to take assay. That is different story. But here different what is that level of bottle, top, middle, bottom of each content uniformity of bottle should be evaluated. So easy of dispersibility. So it is nothing but reconstitution time. Okay, so I will give one video. After that, we can come to these questions. So it will be easy for you to understand. So I just want to show you people. So I hope everyone are there. So I don't know if someone went for slept. So please, sorry, 1 second. So please view this video. One second. Hope is it visible? Hope is it visible to everyone?
No, sir. No, sir.
Now? Is it visible right now, my screen?
It is appearing like N show, sir. That's it.
Yeah.
How should you prepare and administer antibiotics? Oral liquid antibiotics are commonly used among children and infants to treat a variety of bacterial infections. All oral liquid antibiotics vary in their preparation, administration, and storage. This video will show you the general instructions on how to prepare, administer, and store them. Do refer to your antibiotics patient information leaflet or pharmacist for more specific information. Please follow these step-by-step instructions carefully when administering your oral liquid antibiotics. To begin, remove the bottle of dry powdered antibiotic from its packaging. Shake the bottle to loosen the dry powder. Open the cap of the bottle. For antibiotic bottles with markings, pour cool boiled water into the bottle until halfway below the marking indicated on the bottle. As for antibiotic bottles without markings, please refer to the antibiotics packaging, patient information leaflets provided, or your pharmacist for information on the required volume of water to mix the dry powder antibiotic. You may use a marked measuring cup to measure the required amount. Do not use any other liquid to mix with the dry powder antibiotic. Replace the cap of the bottle. Shake the bottle thoroughly to mix the suspension. Ensure that there are no lumps in the mixture. Pop up the remaining amount from the marking of the bottle, or as suggested. Replace the cap of the bottle and shake it well for the second time to mix the suspension. Shake the prepared suspension well before administering. To make sure you are giving the right dose, use the measuring tool that comes with your antibiotic, such as an orange syringe, measuring spoon, or a marked measuring cup. If your antibiotic does not come with a measuring tool, you may ask your pharmacist to recommend one. Do not use a kitchen teaspoon or tablespoon to measure the antibiotic, as they are not accurate. Do note down the preparation and discard date of the antibiotic suspension, and always remember to complete the course of the antibiotic as stated. Store the dry powdered antibiotic at room temperature in a cool, dry place below 25 to 30 degrees Celsius unless otherwise stated. After mixing, store the prepared antibiotic suspension in the refrigerator unless otherwise directed by your pharmacist. Never freeze the preparation. Discard any remaining prepared antibiotic suspension after the completion of the antibiotic course or by the expiry date stated on the label of the bottle. Do take note that some oral liquid antibiotics, such as azithromycin, clarithromycin, and ceftriaxone, have different preparation, administration, and storage instructions. Always remember to consult your pharmacist for any queries or additional information regarding your antibiotic. Thank you.
So everyone have gone through the video, right?
Yes, sir.
So can you any summarize like 2-3 points which you observed in the video? So which can which we feel which is critical.
Antibiotics preparations must be different, sir, not at the same preparations all.
Okay. So different formulations have different reconstitution volume, different storage condition, different usage period. So everything will be different. That's I understood. Next. Anything else you feel which is very critical in that video?
We have to use antibiotics rationally, sir, no?
Sorry, I'm not able to clearly hear that. What is it? So few things I just want to brush up here. So what is the few things is first, whenever you take antibiotics, she was doing loosening of the bottle. So loosening of the bottle is nothing but air entrapped into the blend will be removed if you do loosening of the bottle. If you see, that is the first step what she did. So after doing loosening, she removed the cap and she see is there any marking for the volume to be added. So sometimes they'll give marking, sometimes there will be pill, this much amount of ml of water to be added. So if suppose if it is in Europe or Canada, as pharmacist is going to reconstitute the suspension, they will have measuring cylinder. But suppose if it is in India, we don't have any measuring cylinder in my home. So we'll give marking. So if marking is there, we can directly pour water. Okay. So that also she was saying it should be boiled and cooled water. It's not like normal raw water because chance of microorganism growth will be there. So it should be boiled and cooled water. It's not hot water also. So it should be very clear the terminology. So boiled and cooled water she was adding, and she was saying we should not use other solvent other than water. So not like mango juice or some coconut water I can add, nothing like that. So it's only water to be added because that is only having compatibility. There are other products like cholesterol where they used to take even with ketchups, even with mango juice, everything. But that is having evaluation study done with other vehicle to be administered. But in this case, yes, we have to use only cooled water. Okay, now third, after shaking, if you see, she added exactly half quantity of water and she shaken for some time. So this shaking also matters. So if I say this has a 1 rotation, so now 2, 3. So this number of rotation also I will evaluate during my development. So why I am evaluating is time or number of revolution. So time is nothing but I will take a watch how much seconds after pouring off water I will shake 1, 2, 3, 4, 5. How much time it takes? How many number of revolutions it takes? And some people will shake very slowly. Sorry. Some people will shake very fastly. So what is the shaking mode? So it should be uniform enough, and it should first thing it should be what it should do. It should disperse quickly. If that is not happening, your formulation will not be called as a good formulation. Whatever the case, it should be having a after adding water, after maybe 1 or 2 minutes, it should have effective reconstitution time. That is nothing but reconstitution time. So she added half the quantity of water. She shaken. She just showed us back there is no lumps. So whenever there is no lumps, she added remaining amount of water. So what happens if you add water totally at initial stage? So for example, 100 ml. So they're asking to add 60 ml. So total 60 ml to be added. So I added 60 ml. I'm just shaking. What will happen if in that case? Now instead
uniformity doesn't
evaluated. Now that will not come in during. So anyway, that content will be there. So what will happen now?
Medication, sir.
Sorry, tell me. Sorry.
Regarding to the medication, it does not properly dissolves.
Maybe yeah. That is actually one reason. I'll just give a simple thing. If you think this is the bottle, hope it is visible. Okay. So till here my medication is available, and I have to add water till here. So if you add water till the top, that is the total volume, the head space is very less. The head space is very, very critical for solubilization of powder. Head space is nothing but empty space. So that is why if this powder is there, they're asking to add water till here. I will add a half. So what happens if you add half? Head space is 4. What is the use of head space? So which will be easy or it will be good to shaking. While shaking, it will have more space so that what will happen? It will have the movement, free flow movement, and it will undergo water, convert into a suspension. So that is why we have to ensure that we have to add some amount of water and then shake it properly and see without no lumps, and then you make up the volume to the required level. Or otherwise, first only you add it till the required volume and you are trying to shake it, but because of less head space, okay, there will be some amount of material which is left as a lump. In that case, as she said, uniformity will not be there and it will not have a good, the dose will not come properly outside, and still the end of the preparation, some amount of API will be remained in the formulation. So this is all nothing but demerits. So whenever the simple reconstitution only having this many steps: loosening of powder, removing of cap, adding of boiled and cold water, half the quantity of the water to be added, shake it properly, shaking time, number of rotations you are shaking, and then remaining quantity of water added. Again, you are going to clock it, how much time it, okay, everything is okay, and then you are going to check there is no lumps. So these are the critical steps which you followed, which is very, very critical for a PFOS. So on that behalf, what is the reconstitution time? You have to evaluate critically in the development. And method of reconstitution, I said, what is the mixing speed? You are going to go with respect to time, or with respect to number of revolution, or you are going to say shake till it becomes powder. So three things you can do: shake till it becomes powder, or no lumps are available. So you can give that terminology also. Reconstitution volume and deliverable volume. So this reconstitution volume is always plays critical role in the evaluating the deliverable volume, which is in turn goes to evaluate the number of dose available in the bottle. If I required 80 ml, you mistakenly added 70 ml. Okay, suppose I need 80 ml of water to be made into 100 ml. You added 70 ml. What will happen in this case? Will there be any side effect? Will there be any problem in the patient?
Maybe, sir, some extent it may be affects, sir, because water acts as a solvent, sir.
So here what will happen, right? If you add, instead of 80 ml, if you add 70 ml, only problem is your dose will not be 125 mg. So if I say 20 dose, my each 5 ml contain 125 mg. So when I add less amount of solvent, instead of 80 ml, I add 70 ml, my dose will be around 135 or 132 mg per 5 ml. So the dose will be more. So after taking more dose, what will happen? For example, 20 dose should be available per bottle. So maybe 17 dose or 16 dose will be available. Remaining 4 dose is not there. Because why? You not added the extra amount of water which is actually required. Instead of 80 ml, you added 70 ml. So that is also will lead to what? Improper label claim administration and your dosage duration. Doctor is asking to take for 10 days, 2 doses per day. You will not be able to take on 8th day, 9th day or 10th day because you are not reconstituted properly. So that is one of the causes of reconstitution volume and delivery volume is very, very critical. So flavor evaluation is nothing but, as we said, one of the ingredients is flavor, which we added in the formulation. So how we are going to evaluate? So now the agency is asking us, you added some flavor. As we do PAT, so this flavor at initial as well as end of shelf life. If you say 24-month shelf life or 36 month, during end of shelf life, you have to evaluate the flavor present in the formulation. Maybe it be IR method or very HPLC method, whatever it may be. But it should be very specific to the flavor what used in the formulation. Yes, the flavors what are added in the formulation is still, it is there in the formulation, even in initial formulation stage or end of shelf life, means end of expiry date. During the end of that, the study again will be conducted and will evaluate to say that yes, flavors are there in the formulation. Deliverable dose is linked with the reconstitution volume added, and deliverable volume is nothing but delivered dose. Dose is nothing but 20 dose. So each 5 ml is 1 dose. So 20 dose means 100 ml should be there. So the dose is nothing but 20 numbers is nothing deliverable dose. Uniformity during powder filling. It's very, very critical as we explained. So the filling stage, mg per, if 30 gram per bottle is the fill weight per bottle. If you're not filling it properly, that will also will cause variation in the reconstitution time, reconstitution volume, deliverable volume. So the uniformity of powder filling is also critical, very, very critical in the PFOS, or the actual dose of drug will not be administered to the patient. So this is how the reconstitution volume will be evaluated. I given a simple flow chart. So I'm taking 10 bottles. Okay, I'm adding the required quantity. I'll give an example. I need to add 80 ml. After adding 80 ml for a bottle containing 30 gram of solid, the final delivery volume is 100 ml. This is the concept. So here I have taken 10 bottles. I've checked all 10 bottles. I've added 80 ml. Taken a measuring cylinder added, and then so all the 10 bottles what is the volume obtained? Not less than 100% LV. Lower viscous. So I'm saying low value or lower viscous values minimum 100. Then the test passed. No issue. Okay. If it is less than 100, again, if it is less than 100 also no issue. Volume of one container is above 95. Okay. And then the test fails. If volume of no container is less than 95, means all are above 95. Then again you take 20 more bottle. Okay. So for out of 30, so less than 100% LV, if it is less than 100% lower value, then it is fails. And then if it is not less than 100 LV, again we have to go with this flow chart. So reconstitution volume is also having general chapter as per USP, and we have to evaluate, and it is a very, very critical, which depends upon deliverable volume, which will be affected because of reconstitution volume added. So it should be critically evaluated for a multi-dose container. So that's what. So this is a decision scheme for multi-dose unit container. So AV average volume and this LV labeled amount, labeled volume. So by this, I just want to conclude the dry syrup preparation is suitable, and not only suitable for pediatric, it can be also given for adult, because most of the days now easy of formulation, people are going towards easy formulation or easy of administration or rapid onset of action. So nowadays we are acting on even sustained release suspensions, which can given easily in the form of suspension, but it goes in the stomach and swells and slowly releases the API for a period of 12 to 24 hours. So we are working on that. So it is sort of different formulation attracts always people, so that it will be easy to take by the patient. And particularly dry syrup preparation is advantageous because it is easily weighed, so it can be easily transported as already said, and it is having good stability. So stability is the most problem we are facing in our day-to-day life. So if it is assay and RS is not stable, I'm not going to make sure that my product is good. So by PFOS method of evaluation, it is easy to make sure that it is stable both in dry powder as well as in the form of reconstitution suspension. So some reference I have quoted, so which I have referred some books. So thank you for this opportunity. So I welcome at least 5 to 10 questions from you people, so that I can make sure that someone has learned something new from today's topic. Thank you for one. So is everything okay? Anything questions are there from your end? I can explain it. Hello.
Yes, sir.
So
Hello, if you are having any questions, you can ask for Sir Ma.
I think so. All B Farm and M Farm students are by calling for a meeting like this. I want more people to talk, but here more people are very silent. So that is not going to be useful for either them or me. That's what. So at least you can ask some questions, so where I can feel happy that you learned something new today. So can I call some names so that they can ask some questions? Is it okay, madam?
Okay, sir.
No problem, right? So because I'll just check, is there people are there? So I'll just call by the name what is mentioned over here. Is there Sai Deepika? Is there Sai Deepika?
Sai Deepika. Sai Deepika, just unmute your audio.
See, most of the people are just joined and they left. That's it. Rishi.
No, sir.
Yes, that is
actually here most people are family students. They are having, sir, now.
See, student is a student. Forget about Farm D, B Farm, M Farm. Okay, student is a student. What I used to do in my college days, hey, just give a attendance and just go away like that. Student is a student, right?
They'll ask the questions, but they're.
So let me ask now. Okay, let them ask. So Manasa Naidu.
Sir, myself K. Sushmita, sir.
Yeah, thank you. Please ask your question, ma'am.
Okay.
Sir, you explained very well, sir. So that we are not asking any questions, sir.
So this is to 100% fault. So this is like I'm not accepting this. See,
really, that's true.
Explaining is okay. I accept. Okay, I know something. I explained. but you have to have some doubt now. So now you are a PharmD student. Tomorrow you are going for rounds, means you are going for a hospital. Okay. So if you got a medication, doctor is asking, patient is asking you, Amma, this PFOS, I got it. You just give me how to take the medication. Like that, if any question required or asked by patient to you. So how to tackle the conditions. This is nothing but the pill will be very, very useful. So patient information leaflet. So any question is there, you can ask me, because I'm more related to formulation, not with respect to. So you can ask me with respect to this. Anything is there? I can.
Sir, RPM will affect the manufacturing, sir.
Obviously, na. See, blender is nothing but octagonal blender or OGB, whatever is there. Number of contact points will be there. OGB or what is it, bin blender or whatever it may be. It's a contact point. So example, number of contacts for more, we can ensure more the uniformity. So I think so for bin it is 6 and OGB it is 8 contact points. OGB is octagonal. So 8 points will be there. So here, during my manufacturing, I used to do 12 RPM or maximum 18 RPM I used to go. That is 5 liter to 25 liter capacity is there. But my validation batch, I used to take 6,000 bottles. 6,000 bottles around 100 kg. So that 100 kg, what is the maximum RPM? 6 to 8 RPM. So the machine with respect to its capacity is increasing, RPM will be reduced. Why? So the more the capacity, the lower the shear, higher the efficiency, means higher the effect. Means what? So in R&D, when I do 18 RPM when I keep also, I will get uniform blend, because the quantity is less, machine size is less, uniformity will get. But when you go to increase in the size from 25 liter to 500 liter capacity, 600 liter capacity for a 100 kg blend, the RPM given in the manufacturing, the equipment or handling only 6 to 8. Why? I cannot go more than that, because the more the bigger size, the slower as possible, the more the efficient mixing. So for this, we have to evaluate the RPM of the blender, or otherwise it's very, very difficult. RPM is nothing, blending is nothing but I'm just putting some material in the blender and just mixing it. How much time I want to mix? X question. How much RPM I want to mix? Y question. Both X and Y, if it is in single line, I will get a blend of uniform assay. If not there, for me 3 content will be there for amoxiclav PFOS, amoxicillin, clavulin, potassium, sodium benzoate. If all 3 not coming 100%, okay, so amoxicillin will be 125 mg, clavulin, potassium will be 31.25 mg, sodium benzoate will be 10 mg, which is less in this. 10 mg is less. So my blending efficiency will be declared based upon sodium benzoate, not amoxicillin. Understood? Okay, so here sodium benzoate, if sodium benzoate is uniform, remaining 2 contents will be uniform. So they will evaluate the content which is very least. After blending time optimization RPM, we'll take 10 different location samples and then one composite. We will evaluate for blend uniformity assay, and then we'll say if it is in the range of 95 to 105, it is very good.
Sir, in your manufacture, you didn't explain about glidance.
Lubricant.
Excipients like.
Yeah, actually, PFO is already a powder form. PFO is already a powders. Powders will not have more flow issues. Okay, one thing. And second thing is nothing but during this one, what is that? Bottle filling is there, na? So a hopper will have some vibrators. So this hopper, whatever is there. So we used to go to that floor mill, right? Floor mill for going for floor mill for grinding rice and everything. So same like that, the hopper will have vibrator. So whenever you put the material, it will have always flow capacity more in the vibrator. So very less frequently we use lubricant and glidant in PFOS, because as such the material only powder, it will have good flow property. That is the reason we are not suggesting to add any lubricant or glidant in the formulation.
Thank you, sir. Sir, but you told about the potassium clavulate, it affects on the temperature, regarding to the temperature. Why, yes, sir? Because
the API is not stable. The nature of the drug substances, if it is come in contact with heat. So, normally some API will store at room temperature. Some temperature API will store in fridge. Some API will store in 2-8 degree. So now potassium clavulate, how we will store it? We used to keep in 2-8 degree. Why? If you keep in normal condition, it will start degrading. Assay will come less. And because of that, potassium clavulate will not be dispensed as such. If you take 100 gram of potassium clavulate blend, in that 50 gram will be xylide. Xylide is nothing but silica. Silica is nothing but absorbent. 50 gram of silica, 50 gram of APA. Because the manufacturer only knows this problem. If I potassium chloride, if I keep as such, it will absorb moisture and undergo degradation. So during manufacturing only, he will make a big batch, 100 kg batch of potassium chloride, 100 kg of xylide. He will mix together. So now 200 kg contains 100 kg of APA. So if any sending to us also, it will be diluted, 1:1% diluted, or 50% diluted form of APA. So by this way, exposure to the moisture is reduced. And next storage condition is asking us to store at 28 degree, because room temperature will start degrading. If we start degrading, our assay will not come. Assay is very, very critical for any formulation. That is why it is having, the AP nature is having sensitive to temperature and moisture.
Sir, which validation methods we use in this process, sir?
So here it's not method validation. Method validation will be done for the analytical method. So here I'm saying validation batches means, now I'm saying example. 5 ingredients are there, passed through 30 mesh. Okay, passed through 30 mesh. After 30 mesh, we are putting it in blender. In blender, I'm going for 6 RPM. In 6 RPM, I'm going for 30 minutes. After 30 minutes blending, I'm going for filling. In filling, there will be speeds. Bottle filling, like tablets are there, RPM. So here bottles per minute, BPM will be there, bottles per minute. So 50 bottles per minute, 100 bottles per minute, 500 bottles per minute, like that. So more the faster, more the fast the flow. So here what will happen? This evaluation we used to do each batch. So we have a protocol, process validation protocol. So this process validation protocol will be helpful. My process is robust or not. What is the process? Now sifting, blending, bottle filling, and evaluation, and putting in stability. So this process is nothing but validation batch. So if all the process, 3 batch I'm taking, out of 3 batch, I'll do 2 process validation. So if this 2 batch is coming consistently good, what you will say? Sir, your process is good, sir. Your manufacturing flow is very good. So there is no segregation. So no problem of blend. Everything is good. So that is nothing but validation. This is my formulation validation batches. So method validation is nothing but that will be taken by analytical team before my EB batch. So they will go for method optimization. Partial validation initially they'll do. After that they'll do method validation, and they'll say my method is very robust. If you say 1 mg is there, I can show 1 mg in the formulation. If you say 10 mg is there, 10 mg in the formulation. Like that they'll do method validation. That will be taken care by analytical team. So now whatever I'm talking about is nothing. Validation batch is nothing but process validation. Process is nothing but all my manufacturing process is okay or not. That I am validating by having a process validation protocol.
Sir, you told about the granules, sir, but you didn't mention about the steam process.
So granules is nothing but same sifting we'll do, and then we'll load into that. What is that? After sifting, we'll load into that RMG granulator is there, na RMG granulator. So we'll be loading into that, and just we used to granulate in the form of granules, and that granules will be drying, and then after drying we used to mill it and take a suitable particle size, and then we used to go for mixing with other excipients. So all the steps are there. So I just given brief introductions. Sifting and blending is nothing but only just two process. So granulation, when it comes, we have to form a granules, drying, milling, again as a particle size reduction, and then extra granular addition, again blending, and then go for bottle filling. Is it okay?
Yes, sir.
Okay, thanks. So
Is there any queries, ma? Hello?
So your participant level initially 72, now 48.
Yes, sir, it is going to decrease.
Next, if I ask one more question, I think so everyone will escape.
If you can't ask, you could just message me. I will ask to sir. No, there is no problem.
The problem is what na? Normally we used to go to office by 9 o'clock and used to come by 8 o'clock. 99% we don't have any preparation time, and I will not have any time to talk to anyone, academic persons also. So Sunil somehow he got me, messaged me, troubled me, and somehow he got you take sir like that. Yesterday I had my night shift. I came morning 6 o'clock. So even then also I want to have it done, because it's very less we used to get opportunity to talk, and nowadays there is no chance of traveling also. It's always good to have people's meeting and having a meeting like this. Okay, madam, no issue. I think so.
Okay.
Everyone is feeling very shy. So no need to shy. So you can ask questions, or we can have every month like this any sessions. Or if you want any topic, you can ask me. I will prepare some few days and I can post. I can present it No issue, madam. Like this, we can have some.
Okay, sir.
No problem. If you say, sir, I don't know about Form D, but formulation wise, anything required?
No, sir, they are also having these formulations, all topics.
I
They are
know.
also
Form D
having.
is nothing but B Farm and M Farm together. Only pharmacy practice will be included in the
Here,
Form D.
all students are there for presentation today.
Okay. So no issue. That is good always, but all peoples are there and they should talk. So asking more questions will always make sure that they are learned something. That is what I am saying. Everything is good, no issue. So if anything specific topic required also in future, we can take it up and we can have this. What is that? We can have this
Webinar.
presentation.
Okay,
Yeah.
okay.
Sir,
Okay, carry on. Ask, ma.
I heard some.
Go ahead.
Yeah, tell me, ma.
Sir, antibiotics growth regarding to the manufacture, it differs, sir. How like it differs, sir? In which point of view?
No, your question.
Preparations, sir. Preparations. How preparations? If potassium clavulate like that, sir. Azithromycin, antibiotics, sir. Different antibiotics preparation.
Okay. So now you are talking about that video. In the video, last one word was there. Different antibiotics you have to take in different way. That one you are talking, na?
Yes, sir.
Correct. So in the video, there was last few lines. All the dosage from PFOS should not be taken in the same way. It is different mode of administration or different storage. That is what you are asking right now, correct?
Yes, sir.
Okay. So that is not talking about manufacturing. That is talking about route of administration. Example, I'll tell. So I have till now worked 2 different PFOS projects. One is amoxiclav PFOS. Next, amoxicillin PFOS. When it comes to amoxiclav PFOS, as pot clav is sensitive to temperature and moisture, we should store only 10 days at 2 TA degree only. Means what? I have to keep in fridge only, not outside. Okay. But the same when it comes to amoxicillin PFOS, same there is amoxiclav, potassium clavulate. So that is kept in fridge only. But amoxicillin PFOS, that when I come to that chapter, that reference product is saying 7 days if you keep outside, you can use it. Or if you want to use more number of days, keep in fridge for 14 days so that you can use the 14 days period of time. So the storage is up to you. I don't have fridge in my home. I'll keep RT, room temperature. No problem, 7 days you can use. No, sir, I have fridge in my house. Okay, keep in fridge, use for 14 days. But that is the last day. 7 days means 7th day is last day. 14 days, 14th the last day. And this one thing. And with respect to API, azithromycin or any other product, reconstitution method. Sometimes you will add water first and then mix it, allow to some time for swelling, and then remaining add, then mix it. So the process of reconstitution procedure, water will vary with respect to dosage form to dosage form. So that's what they are saying. Azithromycin or whatever different formulation you have, the mixing way, mixing speed or mixing volume will be different. Storage condition also will be different. Something will keep in RT, something will keep in fridge. Okay, so this also will vary. So on that is what they're saying. We have to cleanly study the pill, which will give what is the volume to be added, how you have to mix it, how much days you can keep, what is the storage condition. This 4 information will be clearly mentioned in the pill. So all the dosage form are beta-lactam antibiotics only, but everything will vary with respect to one another. Understood, na?
Thank you, sir. Thank you for your valuable information, sir.
Okay, information is always valuable if you use in your life. Thank you.
Sir, I do have a doubt, sir. Actually, the doctors will say after opening the containers, pediatric, based on the pediatric children's, after opening the syrup bottles, after 1 month, we should not use like that, they will say.
See, again, that's a problem with you people. I'm saying now, see, doctor is not a, doctor is only a prescriber. He can just write, he is having this disease, so for that he can give so and so medication. No, you are a pharmacist. You know everything about the formulation or a product which is there in your pharmacy. So if you take the pill, patient information leaflet, or you take the label, you study it, there only it is given. If doctor says it is no, he is not a god. He just knows how to find out the particular disease in the patient. That he is well versed. But we know what? More than him. For this particular disease, which medication will be more suitable, that we should know. And we should tell to the doctor. So what you say, what I'm saying to you, it is not doctor what he says to you. I'm saying to you that in the dosage form, in the label claim, in the label or in the pill, you have clear information how to store the medication. If you open the bottle, as you said, if 1 month is given the pill or the label, yes, 1 month you can use. If it is not given, if we given only for 14 days, and if doctor says 1 month, will you use it for 1 month?
After opening that bottle,
Opening the bottle,
after
label it is saying 14 days, but doctor is saying to use for 1 month. Will you use?
No, sir, it is not about 1 month. For children, we should not, after opening that bottle, they will say for 7 days medication. After completing the 7 days medication, the medicine will be left, some medication will be left in the bottle. But we should not use that left-handed medication after 1 month, they will say.
Correct. Not after 1 month, ma. After that 7th day only you cannot use.
Okay.
See, understand the question. See, antibiotics are not stable in aqueous formulation. Means what? When it's come in contact with water, my API starts degrading. So what I am giving? My assay limit I am giving 80 to 120. 80 to 120 means what? If I think like 125 mg is there, okay, dose is 125 mg. Out of that, what is the 80%? Will it be very less? So on 7th day or 10th day, the assay of my formulation will be around 80%. Understood, na? So yes, what is happening after reconstitution? 7 days you can take. So if the pill only saying 7 days you have to take, after 7th day what happening? 8th day, the assay slowly will come down. After 7th day what happened? Assay is coming down. There is no therapeutic use. So on 8th day onwards what happened? No use of taking the medication. So will you take anything which is not useful for you?
No.
So that is why the doctor is saying after opening of the container, after 7 days, whatever the remaining, forget about you using or not. You reconstitute and kept it. You not used also, no problem. You have to throw after the end of that particular number of period. It is 7 days or 10 days or 14 days as per the pill. You have to store it properly. After the 7th day, 10th day, 14th day, you have to throw it. Forget about 1 month and all. I'm not saying 1 month. I'm saying as per pill, if it is 7 days, 7th day throw. 10th day means 10th day you have to throw. That's it. Understood, na? So after 10th day, what happened? The assay will degrade slowly from initial to 10th day. It's not on 10th day, not only on 10th day. It will be from starting from day 0 to day 10. But it will be in the range, assay limit range. But after 10th day, what happened? It will go below the limit. So what happening? There won't be, effect won't be there if you take the medication. Or effect will be there to a lesser extent, not to the expected range. So that is the reason we should not take after that particular days.
Okay, thank you very much, sir.
Large amounts of antibiotics may cause resistance, no, sir. Using large amounts of antibiotics may cause to resistance, no, sir.
Okay.
So that we are using to some extent, sir. If doctor says use for 7 days or if anything problem to us, sir, if it relieves for 1 day, we doesn't use, no, sir, tablets.
So now question is 2 things, sir. One, you don't want antibiotic resistance in your body. So for that, you want to use less number of days. That is the first question. Second, what I want to say is, now doctor is asking you to take for 1 week. That is 7 days, 2 doses per day. Okay. Now you have cold, cough, fever, everything is there. You taken for 4 days. Okay. After taking for 4 days, you got a good relief. Everything is there. You got normal. Everything is okay. So now you are skipping your 5th, 6th, 7th day dose. As you said, you are skipping your dose. Now, after 7th day, maybe after 1 week, again you are getting fever. And then you are going back to doctor. Doctor, I will ask you, Amma, I asked you to take for 7 days, 2 doses per day. Have you taken it properly? No, sir, I have taken till 4th day. Everything was good. So I stopped on 4th day because of antibiotic resistance. Now he will ask, so we don't know about antibiotic resistance. Everyone is having the problem of antibiotic resistance, but recurrence chance means what? The virus or bacteria should go away from your body. So it should completely be removed. That's the reason they are saying for 7 days. So once you taken for 7 days, recurrence chance is less. So that is why they'll give for 7 days or 10 days. And that also, most of the beta-lactam nowadays, we are trying to reduce it, usage. So if you feel if it is okay with paracetamol, you can take it and we can resolve the issue. No, sir, I already have a lot of issue with my health. It should be as quick as possible. It should be requirement. Then you have to go with antibiotics only. So we cannot skip. So you cannot say here given for 7 days, I'm going for only 4 days because to reduce the resistance, that is not possible. Here the thing is about curing, complete curing. Is it okay? I'm able to answer your question?
Yes, sir. Thank you, sir.
Is there any queries, ma? Okay, sir.
So is it okay? Can we end it up?
Yes, sir. Indeed, it is a great honor and privilege to propose a vote of thanks for me, sir. First and foremost, I'm very thankful for you by disputing your busy schedule to found time for this national webinar and giving such a great, valuable, informative, interesting, and communicate talk on these powders and oral suspension consideration for the regulatory affairs of this market. Thanking you all for all participants for your active participation. Hopeful the presentation is beneficial, profitable, and fruitful to everyone, to all of you. Thanking you one and all. Thanking you very much, sir, for your valuable time and for your valuable
Thank you very
speech.
much. And you also make sure that we are getting like good topics, so we can have a discussion like this every month. So there is
Yes,
nothing
sir.
busy schedule. I'm trying to put my effort to study pharmacy, what I studied at. You are putting your effort to give a good student to the industry background. There is no one is busy in this life. So I came from there. So I'm giving it back to you people. So I want you people to have a good discussion on good topics, whichever you feel it is good, and you can discuss and come up again to me, and I can make sure that I can help you to my,
Yes,
my best. Okay.
Okay, okay, sir.
Okay, ma'am.
Thank you very much, sir.